Welcome students, welcome to Zoology Wala, and today in this session I am going to explain you about the physiology of digestion. In the previous sessions I explained all the structural components and digestive juices which are produced in the alimentary canal, and now we will see how those enzymes will assist in the functionality of the body. I mean to say, in order to break down the complex food materials into simple food materials. That is the main part of digestion. That's why we are discussing the digestion. So first of all, the saliva, the human, see the digestion begins in the oral cavity or buccal cavity itself. There are two types of digestions are present, I mentioned in the first class of the digestion. Mechanical digestion and biochemical or enzymatic digestion. Mechanical digestion is simply mechanically the food material will be crushed with the help of teeth and with the help of the peristaltic movements of muscles. The food materials will be slowly passed from one part of the alimentary canal to another part of alimentary canal, and food material is also broken down into small food particles. That is mechanical digestion. And now the enzymatic digestion, it begins with the help of enzymes. So the mechanical digestion and biochemical digestion, both digestions will begin inside the stomach itself, inside the mouth itself, and the food material, basically the starch what we are taking, will be digested into maltose. Nearly 30% of the starch what the human beings will take is converted to maltose in presence of an enzyme, salivary amylase. Salivary amylase, also called ptyalin, I already told you it works at the pH 6.8. In the NEET aspect, you will be asked with so many questions. You will be asked with so many questions, something like which of the following enzymes will work in which medium, or for the digestion of proteins, which medium is required. Digestion of proteins will begin at, digestion of proteins end at, name of the substrate, name of the enzyme, pH. Like that, number of questions will be asked. You should be very cautious. So the starch is converted to maltose in presence of salivary amylase in pH 6.8. If you observe the stomach, in presence of an enzyme, in presence of so many components, and especially in presence of HCl, hydrochloric acid, pepsinogen, which is an inactive enzyme, pepsinogen is converted to active enzyme pepsin in presence of HCl. Pepsin plays an important role in the digestion of proteins. Okay, so the proteins which are present inside the stomach are now converted to proteases and peptones in the stomach itself. So in the stomach itself, the proteins are broken down into proteases and peptones, where the number of amino acids are comparatively less in proteases and peptones when compared with the proteins. And this type of formation of products will take place in presence of the enzyme pepsin, for which HCl action is very much essential. So in presence of HCl, zymogen products, zymogen cells, I mean to say, inactive enzymes are converted to active enzymes in presence of HCl. And of course, prorenin is a very important one for curdling of milk. In case of infants, prorenin is also converted to renin, but that renin is, which is a milk curdling product, it is seen in infants. That is the reason why it is advisable to take milk, especially to give milk for the young ones. In case of adults, that milk curdling product is generally not seen, the so-called renin. That is the reason why it is not that much advisable to take the pure form of milk for adults, and of course the pure form of milk is not advisable even for infants also, and that I will tell in another context. The proteins are converted to proteases and peptones, and now the proteases and peptones which are formed in the stomach moves gradually into the duodenum, but here the protein digestion is not going to be finished. Protein digestion just began inside the stomach. And if you move to the next one, you see here the proteins, proteases, proteins. What are these proteins? You may ask me, sir, already you have written the proteins. Proteins are converted to proteases and peptones. Okay, but they are nothing but the proteins which were converted to proteases and peptones in the stomach. Proteases and peptones which moves into the duodenum, and the native proteins which are present in the duodenum. Native proteins means the proteins as such which release into the duodenum. Why? Because the stomach is not going to handle all proteins. So the native proteins, proteases and peptones which are coming from the stomach or which came from the stomach are going to be converted into dipeptide in presence of these enzymes like trypsin, chymotrypsin, carboxypeptidase. See, trypsinogen is an inactive enzyme. I already told in my previous classes. Trypsinogen is an active enzyme, is converted to trypsin in presence of an enzyme activator called enterokinase. And this enterokinase is an activator molecule present in the intestine, and this enterokinase activates the trypsinogen to trypsin, and now the autocatalysis also will take place. I mean, the produced trypsin will act on the remaining trypsinogen molecules, and all the trypsinogen molecules are converted to trypsin. Please listen carefully. Some trypsinogen molecules are converted to trypsin in presence of enterokinase, and the produced trypsin acts on the remaining trypsinogen molecules, remaining inactive trypsinogen, to convert them into active trypsin, which comes under autocatalysis. Not only that, the trypsin plays a very important role in conversion of remaining inactive enzymes which are present in the pancreatic juice also into active components of pancreatic juice. So, trypsin, chymotrypsin and carboxypeptidase, previously I mentioned procarboxypeptidase is an inactive enzyme which will become active into carboxypeptidase, and they are converted to dipeptides. Now, see here, polysaccharides, starch, already I mentioned in the mouth itself 30% of the polysaccharides are starch converted into maltose, and now the remaining also converted to disaccharides in presence of pancreatic amylase. And here number of fat molecules also present inside the food. I already told you, my dear friends, the stomach or the mouth or the intestine contains different components, among those carbohydrates, number 1, number 2 proteins, number 3 fats. Carbohydrates, proteins and fats. These three are the major components which plays an important role, and these major components, they won't enter into the blood as such. We have to break them. Our body has to break them into simple compounds. Carbohydrate should be broken down into sugars, protein should be broken down into amino acids, fat should be broken down to fatty acids and glycerol. Then only they will be able to enter into the different fluids present inside the body like blood and lymph. So here fats are going to be converted to diglycerides, which in turn converts to monoglycerides in presence of lipase, who is going to active the lipase enzyme, who is going to activate the lipase enzymes. Bile is going to activate the lipase. I already told you that liver secretes bile, a very important component, and bile contains no enzymes, but it contains so many compounds which are very much essential for the emulsification of fats, breakdown of large chain fatty acids into small fatty acids, so that the surface area of the fatty acids will be increased, and further digestion of fats will continue because of the bile. And bile contains phospholipids also, and bile salts are present which are able to attach to the fat globule. Digestion of fats will become so much easy. I will tell at the time of absorption of fats, don't worry. So the fats are converted to diglycerides in presence of activated lipases, which are activated lipases means which are activated by bile into diglycerides and into monoglycerides. And now the nucleic acids which are present in the food material, these are also important components of the food material, and the nucleic acids are also converted to nucleotides in presence of nucleases, nucleotidases, essential. I will show in the next slide. Nucleotides, nucleosides. Nucleotidases helps in breakdown of nucleotides. Nucleosidases helps in conversion of nucleosides finally into sugar and base. So of course sugar, base, phosphates are the three components present in the nucleic acids. So these three components will be finally formed as a product from the nucleic acids. So nucleases, pancreas produces nuclease, converts the nucleotides, helps in formation of nucleotides, which in turn forms into nucleosides. You see here dipeptides. Dipeptides are nothing but the intermediary products of the protein digestion. Dipeptides are converted to amino acids in presence of dipeptidase, tripeptidase, dipeptidase. These are the enzymes produced by intestinal juice called succus entericus. Intestinal juice or succus entericus, these are the secretions of brush border, or simply let me write the crypts of Lieberkon and the Brunner's glands which are present in the submucosa of duodenum. So the secretions put together forms succus entericus. So the succus entericus contains these dipeptidases, tripeptidases, which helps in conversion of dipeptides to amino acids. If at all amino acids are formed, we should feel happy. Why? Because protein digestion is finished. So maltose already we have seen as an end product of the starch digestion in the mouth. 30% of maltose already produced, which is coming from mouth, and now the maltose is converted to two molecules of glucose. It means one glucose and another glucose, two glucose molecules combined, and it is nothing but the maltose. Maltase enzyme helps in breakdown of these two glucose molecules. So one maltose molecule will give two glucose molecules. So in the examination you will be asked for the formation of 100 glucose molecules, how many maltose, how many sucrose molecules are required, like that the questions will be there. From the 100 maltose molecules, 200 glucose molecules will form. So in order to form 200 glucose molecules, 100 maltose molecules, or in order to form 400 glucose molecules, 200 maltose molecules are required. And lactose, in presence of an enzyme lactase, converts to glucose and galactose, a very important protein present in milk also. Sucrose, with the help of sucrase, which is also called invertase, sucrase, in presence of sucrase or invertase, sucrose molecule will forms glucose and fructose. So glucose and fructose combine to form sucrose. So sucrase enzyme breaks the glucose and fructose bond, and so from one sucrose molecule, one glucose and one fructose. Similarly, from 100 sucrose molecules, 100 glucose molecules and 100 fructose molecules will form. Nucleotides, with the help of nucleotidases to form nucleosides, nucleosidases, with the help of nucleosidases, sugar and base finally formed. So nucleosides in turn broken down to form sugars and base. Di and monoglycerides, already we have seen fat digestion already began. You see the fat digestion. So fats, diglycerides and monoglycerides, these are not the simple form of fats. They can't enter into even lymph also. Even though lymph capillary is a lacteal, even though lymph capillary is large, they can't enter. So in order to enter into the lacteals, it means large lymph vessel, di and monoglycerides in presence of lipases, small quantity of the lipases are present in the gastric juice, and intestinal juice contains lipase, pancreatic juice contains lipase, and with the help of these lipases, finally fatty acids and glycerol molecules are formed. So what are the end products of the fat digestion? End products of the fats, finally fatty acids and glycerol. What are the end products of the protein digestion? Proteins finally formed amino acids. And what are the end products of the carbohydrates? Sugars. So like that, these end products are formed, and now these end products are ready to enter into the body. You see, hydrochloric acid is a very important one for the activation of inactive enzymes, but in order to protect that high acidity, gastric juice pH will be around 0.8 to 1.8, something like that. It means on an average you can write 1.0 to 2.0, okay, or 0.8 to 1.8, something like that. On an average I am saying. So 0.9 to 1.8, something like that pH. So in that 0.9 to 1.8, something like that, on less than 7, in that high acidic pH, the stomach walls can't bear that high acidic nature. That is the reason why oblique muscles are already there in the stomach. I already told you the histology. In addition to that, water and bicarbonates are also present in this gastric juice, which helps in prevention of excoriation of the walls of the gastric epithelium. So the hydrochloric acid, though it is present in stomach, the walls of the stomach may get damaged because of the HCl. So water and bicarbonates helps in prevention of damage of walls of the stomach, and water and bicarbonates are already produced by pancreas in order to neutralize the acidic food which is coming from stomach. Listen my dear friends, food material slowly enter from mouth to stomach. In the stomach, HCl mixed with the food material. So now acidity increased, and the partially digested acidic food in the stomach is chyme, chyme. partial digestion only, not complete digestion. I already told protein digestion began in stomach but not finished. That partly digested acidic food slowly enters into the duodenum with the help of the pyloric sphincter. Slowly it enters into the duodenum, but wall of the stomach is able to bear the acidic nature, but the intestinal walls are not able to bear the acidic nature. So immediately when the food material enters into the duodenum, the signals will be given to the pancreas. My dear friend pancreas, food is coming from the stomach that contains very acidic nature, we can't bear that acidic, you do something. Like that, one hormone will give signal to the pancreas. Who is that hormone? Guess? I will tell at the time of hormonal interaction. So pancreas will receive the signal. Immediately pancreas, okay, don't worry, I am there. It releases water and bicarbonates. So the acidic food which is coming from the stomach will be neutralized because of the secretions released by the pancreas. So acidic food will become alkaline. Alkaline nature is very much important for the activation of enzymes which are present in the intestine. So acidic food is converted to alkaline food, then only the functionality of enzymes present in the intestine will be activated. And now what about large intestine? Is it going to play any important role in the digestion? No. Large intestine plays an important role in the reabsorption of water, not only in human being, in any animal. Large intestine of course hosts symbiotic bacteria. Cellulose digestion will take place partly, but that also not it won't contribute much for the human beings. Why? Because much of the food material what human being is taking won't contain cellulose. So water reabsorption and minerals reabsorption, drugs reabsorption, some sort of drugs will be absorbed in the large intestine and some drugs will be absorbed in the mouth. I will tell at the time of absorption, that is a different story. But now all the end products are ready in the lumen, and now the next step, how these are going to enter into the body, absorption, that I will tell in my next video. Thank you, signing off.